In the previous two videos, I talked about lipids and their structures. And now in this chapter, we're going to take a look at when those lipids associate to one another with one another, they're going to make membranes. And so we're going to look at the membrane, what its purpose is, and just go into a little bit more detail on what membranes do. So if we take a look here, there's going to be three major classes or structures that are observed when you take, let's just say, a handful of lipids and we throw them into water. What are three possible ways that they can associate with one another and form some kind of structure here? And so you see there we have the micelles, bilayers, and liposomes, or what's called a vesicle. So if we look at the pictures of these things right here, so if we look at a micelle, so remember what this word right here, epipathic, means, right? That means that we have a polar head or a hydrophilic part of the molecule, something that loves to interact with water. And then you have a hydrophobic piece, a piece that does not like to associate with water. It's too high in energy to do so. And so depending on the lipids, this is what's going to determine or help determine what kind of structure we're going to get here. Are we going to get a micelle, or are we going to get a bilayer or a liposome? and I'm going to have to erase this. Okay. Look at the shape that the lipid is making right there. Kind of has that cone shape. And when the lipid itself has a shape like that, but it's not just I'm misspeaking here. It's not the shape, it is the cross section. Right there. So if you take a lipid and look at a cross section of that lipid, if it has that cone-looking shape right there, that's going to give us a micelle. So one way I'm going to look at it is if you have the polar head and then the tail. What we're simply looking at, what's the distance between that edge and that edge? And we have a distance right there. But then when we look at this edge and that edge right there, you can see it's more narrow, it's smaller. So we get a cone-looking shape. Well, the way this is drawn here, yeah, okay. Now when you have lipids that are, the cross section is more cylindrical, you can see we're going to generate a bilayer. And when we look at that bilayer, we see that there's a top leaflet and a bottom leaflet there, and that the hydrophobic tails, what are they doing? They're associating with one another. So if you take a handful of these lipids and shake them up and you throw them into water, they're going to associate in a bilayer. And so it's minimizing the interaction of those hydrophobic tails with the water. And so we can see that on this side right here, we have the polar heads, and then on the opposite side, we have the polar heads. And it's those polar heads that are interacting with water to form this bilayer. Now we have in this image a vesicle. You can see it also is a bilayer, but now it forms this sphere. And within that sphere, you have an aqueous pocket. Now, why would the lipids do this? Well, if we take a look at this bilayer here, I just explained why we have a bilayer, right? Because we want to get those hydrophobic tails away from the aqueous environment. But look at this. What about that edge and the edge on the other side? If it just formed a bilayer, you have these edges right here that are exposed to water. And so to get rid of that exposure on the edges of this bilayer, these two ends come together and form the sphere. And that's what we get here. So that's another driving force to get these shapes right here. Let's go to the next slide. Whenever you guys see students read in the corner right there, that's not saying this information on the slide is not important. It's just, I feel there's no need for me to just read these bullet points to you. And sometimes it's because this is kind of just a review and you've seen this before in your previous classes. So make sure you read these slides right here. I'm just going to skip ahead to this one right here. Okay. So as you read through this slide here, there's a lot of good information on it. But the main takeaway that I want you guys to get from this slide is the main purpose of the cell membrane is to regulate what can come in and what can go out of a cell. And one way that it regulates this is just by the intrinsic nature of the membrane itself. And that has to do with its chemical structure here. So if we just take a look at the bilayer, okay, so let's say this is the outside of the cell and this is the inside of the cell. Okay, so we have those areas separated by this membrane. So let's see what happens here. What if we have a polar molecule? Let's say polar molecule. We have a polar molecule on the outside of the cell. Is it going to be able to just come through to the inside of the cell very easily? No, it's not. Because remember, from organic chemistry, this principle, like dissolves like. If we have a polar compound and it's in the aqueous environment outside of the cell, it's fine. But that polar molecule, in order to get to the inside of the cell, it would have to transverse through all of this. And what is that? That is nonpolar side chain or nonpolar fatty acid tails. It's also very greasy. So all these things that we can describe in those hydrophobic tails, they're greasy, nonpolar, everything that this polar molecule isn't. And so that polar molecule is going to have a very difficult time to get through. And so that's one way we're going to keep polar solutes out, polar compounds out, is because of this membrane. It's too greasy in the middle there. Or better, or said another way, it's just nonpolar. Okay. And based off the same principle too, what if you have charged species here? Negatively charged species or positively charged species? Those charged species are also going to have a hard time transversing through the membrane. So the membrane itself just keeps things out and regulates that. And as we progress, we're going to say, well, what if we need a polar compound to get onto the inside of the cell? There's going to be some ways that we will discuss how that works. But the big picture from this slide right here is that the membrane is designed to keep things out. But with that, there's the caveat. The lipids in the membrane are designed to keep things out. But then the membrane itself is going to have other molecules, such as proteins and ion channels, and these different types of proteins that allow things to come in and out of the cell. And sometimes those happen passively, where they just happen. And then there's times where it requires some energy to get it to go across the membrane through these proteins. Okay, so I want to make that distinction, that the membrane's purpose is to keep things out, but it also has parts in the membrane to allow things to come in and out, but it's highly regulated. Okay, another student read slide here. Okay, we have something called the fluid mosaic model of the membrane. And when you take a look at this membrane here on this slide, we see the lipids, okay? But look at what we're showing also. We have the green ones right here. Those are lipids, but those are the sphingolipids, like we talked about in the previous videos. And then we have the blue ones right here. We have our different phospholipids. And so we also have the top leaflet and the bottom leaflet. And what you're going to realize really soon is that the composition of the top leaflet is oftentimes going to look different than the composition of the bottom leaflet. They're not the same. Within this membrane, we're going to have what's called integral proteins, which span, you have protein on the inside of the cell and part of the protein on the outside of the cell. Okay, and then we're going to have peripheral proteins that are just associating to one of the leaflets. We see here we have some sugars attached to the proteins. We have sugars attached to the lipids. Like we have the sterols, which is basically cholesterol. There's so much that's within a membrane. Okay? And then this picture doesn't have everything that's in a membrane. We have ion channels that we're going to talk about, all sorts of cool stuff, okay? But this fluid mosaic model is demonstrating that this membrane, this lipid, let's just look at this particular lipid right here. That lipid isn't frozen in space. It's moving. These things are moving throughout the membrane. Okay, it's fluid. That's fluid-like. And so I have this video right here that you can watch on your time, and it just shows what I mean by all these pieces right here are just like floating on a sea, just moving around. And so that's what the fluid mosaic model is describing, that phrase. Things are moving around. So if we go to the next slide, we see something here talking about the composition of membranes. And it's going to differ based off of the organism, the tissue, where these cells are found. It's all going to have a variety of what's in that membrane. And so this table right here shows us when we're looking at different type of species, mouse, yeast, E. coli, and so forth, look at the composition of the cell membrane. Just from mouse is 45 to E. coli is 75% of the component of the membrane is protein. And you look at the type of lipids right here. How many phospholipids does it have compared to other types of lipids? Depending on the species, we're going to get membranes that look different. And then what's also cool is within the same species, but when you look at the plasma versus the rough ER or mitochondria right there, those components have a membrane, and the composition of those organelles in cells have different types of phospholipids. Not phospholipids, different types of lipids. You can see right here, the main ones right here, the sphingolipids, the phosphatidylcholine, and so forth. Those three right here, you can see color-coded really quickly and really easily that, hey, within the same species, you could look at in a particular, just the cell, let's say you look at the cells and at the different organelles, they're going to have a different composition. So lots and lots of variety. Okay. Okay, let's talk about this membrane composition some more and this term asymmetry right here. Okay. So in order to understand asymmetry, let's talk about how these lipids are created and where they're created. So the lipids are synthesized in the ER. This is where they start. And so you see this little rectangle right here? It's showing you the composition of those lipids right there. And when you take a look at the top leaflet and the bottom leaflet here, you can see that the colors right here on the top and on the bottom are relatively the same. So that membrane right there, it has some symmetry to it. Right? But when you compare this membrane up here, you can see that the colors at the bottom and on the top are different. And so this membrane right here has some asymmetry to it. And this is really, really important because what happens is the composition of these lipids determines where it needs to go in the cell. It is the identity. It helps to identify what type of membrane or where that membrane needs to go. So when you're looking at this graphic, I want you guys to pay attention to these terms right here. Here's the lumen and the cytosol. So when we come over here, where's the cytosol? It's the yellow right here. It's all that yellow fluid, really. Okay? And when we look at this membrane here, this lumen, where's the lumen? It's on the inside right here. Okay? Now, as the membranes travel to the Golgi apparatus, it's here where they can undergo the asymmetry, where things can get changed around to help determine where it needs to go. Now, when we come and look at this part right here, you can see that we're calling that the cytosol, that one right there. So that would be what? So these lipids right here are on the cytosol side of this green line right here. And then these lipids right here, where are those? Those are inside the lumen. So in this space right here. However, we can look at this same image, but with respect to the outer membrane right there. And so when we look at that version right there, the cytosol is right here, and the extracellular space is over here. That's the extracellular space. And so for this graphic, it can represent this one right here and that one right there. So pay close attention to where these bilayers are found. Okay? And the reason why I'm telling you to pay attention to those things is because depending on where depending on the composition is very important for the lipids function. So for example, look at this here. If you have a lot of the phosphatidylserine, if you have a lot, no, we have to specify where. If you have a lot of the phosphatidylserine on the outer leaflet, this one right here. So with respect to this region right here, if you have a lot of phosphatidylserine on the outer leaflet, it is marking that particular cell for apoptosis, which is just cell death. And so that could be very scary and risky. What if, for some reason, your body's taking and sticking a lot of phosphatidylserines on the outside of the membrane when it shouldn't? Then your cells are going to be dying, right? So you'll have a disease. And so biology has a way to control the composition of these membranes, which is so cool. So the asymmetry, why is it important? Because it determines what type of cell membrane you're going to have. It's going to determine if the cell lives, if it dies, and another host of things that we will talk about later. Go to the next slide here. So I talked about this in my previous videos, but I feel it's really important just to highlight it and remind everyone about this. Okay. So we've been looking at these bilayers, right? There's two, but in some species of Archaea, there's going to be species that live in extreme environments, and that they're not going to have their membranes aren't going to be bilayers, they're going to be monolayers, because we have these types of our ether lipids right here. And I highlighted why these ether lipids are so stable, more stable than the phospholipids. And that has to do, if you recall from that video, has to do with, hey, that is a ester linkage versus a ether linkage. And ether linkages are more stable. Another way that I remember this is when you go into the organic chemistry lab, have you ever grabbed a solvent that was an ether? No? No? But you have gone into the lab and grabbed ethers, diethyl ether, as solvents. And so in my mind, I'm like, if you don't see solvents as ethers, as esters, you don't find solvents as esters, but you do ethers, that implies that, hey, an ether is a good solvent because it's inert. It's not very reactive. So that's just one connection that I make to help remember why is that ether linkage so important? Because it's stable. Okay, I've highlighted about these things before, channels and receptors and all that stuff. We're going to get into a lot of the details of these things right here and why they're so important with respect to membranes. So next, in this slide, we're going to talk about proteins, and they're going to be classified based off of how easy they are to remove and to isolate, and also how they interact with the membrane. So the first one that I want to talk about is peripheral proteins. Let's write that word down. Peripheral proteins. Peripheral proteins are right here. Okay. And peripheral proteins are going to interact with other proteins that are in the membrane or on the membrane by electrostatic interactions. So you can see that there's some positive charges here on this protein, and so there's going to be some negative charges there on that peripheral protein, and that's going to associate with this protein that's in the membrane by electrostatic interactions. These peripheral proteins can also interact with the membrane itself via electrostatic interactions. And so if we wanted to isolate peripheral proteins, because they're only associated to the membrane via electrostatic interactions, we can weaken that interaction by changing the pH or adding in some urea and to disrupt that ionic interaction. And so we could isolate those proteins in that way. And so that's the definition of a peripheral protein. Then we have something called an integral protein shown right here. And an integral protein, there's going to be two classes. We have polytopic and monotopic. And the difference between a polytopic and a monotopic protein is in a polytopic protein, the protein spans both leaflets of the membrane. In comparison to a monotopic protein, it only goes through one of the leaflets. It stops right there, doesn't go all the way through. And so these proteins, if we wanted to isolate them from the membrane, it's going to require a little bit more effort. It's going to require a detergent that totally disrupts the interaction between these two proteins with the membrane. As you can see here, we are adding that detergent right there to rip it out. I think ripping out is too strong of a word, but to get it out of the membrane and to isolate the proteins. And then we have our phosphotropic proteins. Now, the phosphotropic proteins, they're going to be found in the cytosol and on the membrane. So let's take a look at this guy right here, over here. So it can be just free floating in the cytosol, or it can be linked to the membrane via a covalent bond. Sometimes those are sugar linkages, and sometimes it's other different types of linkages. But this protein can associate with the membrane and float away. So it can be in two parts of the cell. And then you have on this side right here, this is also an amphitropic protein in which it is associating with the membrane. Okay? And then it's falling off. Right? And so in this protein right here, how does it know when to bind the membrane? Well, cells regulate this very tightly, and oftentimes it's due to phosphorylation events, which is a way to get proteins to do what we want it to do or to turn it off. So we've talked about that when we're looking at enzymes and whatnot. But kind of the same idea though, phosphorylation can influence a protein to bind to the membrane or not, or vice versa. Okay? So, but this linkage here, this right here is non-covalent, and this is reversible. You can see the arrows right there. So that's a reversible process, whereas this one typically is not when you chop off a covalent bond. So in this slide, I have 4 examples on how we can link a protein to a membrane. And generally speaking, what's happening here is we can have a protein represented by the circle here, and to either you can see here either on the internal cysteine or serine, or the amino terminus of glycine, or the C terminus of cysteine. There's all these different parts of the protein where you can attach a fatty acid. Here are some fatty acids right here. This one right here is a little bit different. That one's not a fatty acid. That's a isoprenoid. But they still basically just this long carbon chain, nice and greasy. And so what happens is that this ball or this protein right here is representing these peach blobs right here. And what's going to happen is we have this protein and we want it to associate with the membrane. So the way that that's done is we attach some kind of fatty acid to it. Okay, there's our fatty acid, or it could be an isoprenoid. So we add a fatty acid. Why do we want to do that to the protein? Because now this protein now can come get close to the membrane, and then this hydrophobic piece right here is just going to insert itself into the membrane. And because that fatty acid right there is going to associate with all the lipids in the membrane, it kind of anchors it there. But when we look at this with some perspective here, oftentimes these proteins are really, really large and a fatty acid chain tiny in comparison. And so really this little linker right here, this fatty acid linker, is not technically going to hold the protein in the membrane very well. It's a good start. But what else influences a protein to associate with the membrane is also how the protein itself, right here, interacts with the membrane. So there's going to be some favorable interactions between the protein and the membrane to help keep it there, and the linker right there. So that's the general idea of what's going on, is we are going to have a protein, we'll attach some kind of fatty acid to it, and then that protein fatty acid pair is going to come in and insert itself into the membrane. Now I want you guys to understand these names right here and what that's talking about. So let's clean this up here. Take a look at what's going on here. So we have the palmitoyl group right here. What's going on here? Well, the image or the slide's telling us here that we could have any type of protein, and what we're looking for within that protein is a cysteine or a serine, and that is going to be on the internal side of the protein. It's not going to be on the N terminus or the C terminus. It's just going to be smack dab somewhere other than those two spots or the two termini. Okay? And so we are going to attach a fatty acid. Which one? Well, the name right there tells us. So if you go back to our discussion about fatty acids, you'll recall looking at a table or in your notes that we have something called a palmitic acid. So what we're doing is we're taking a protein and attaching a palmitic acid to it. And now we call it that, palmitoyl. I'm probably mispronouncing that. Okay. Well, that's what's going on there. Now when we take a look, making sure this is recording, now, what if we have a protein and to the N terminus of a glycine residue in that protein, we are going to attach a different fatty acid. And that will be this guy right here. one, or you misspelling it. Myristic acid. If you take myristic acid, attach it to the N-terminus of a glycine residue of a protein, you get that name. That connection right there. Myristoyl. Myristoyl. Okay, so we have the myristoyl group right there. And so that fatty acid right there and that fatty acid right there are two different types of fatty acids. And then here we have the farnesyl right there. And that farnesyl comes from this isoprenoid. And this isoprenoid looks like this. Let me double check the structure here. I want to not mess this up here. So the structure of that's going to look like this. 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. And of course, we have to have something like that. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. And then we'll attach that to an oxygen. And we're going to say 2 phosphates. I'm not going to draw out the phosphates. Okay, so we have 2 phosphates attached here. And then we would have a double bond there. And so that isoprenoid is called the So this particular molecule is the farnesyl diphosphate. Okay. And what's going to happen is that the C-terminus of a protein, there's our C-terminus right here, but we are going to have what? A cysteine. So there's our cysteine. Now what's going to happen is that that sulfur atom right there is simply just going to come in and form a bond with that group right there. And that's why you have this thiol right here, that thiol linkage. And this linkage right here, we would classify that as what? A thioester. So we have a thioester. Okay? We'll get a thioester here when cysteine is the amino acid. When serine is used, what do you think the functional group is going to be right here? What type of linkage if it's a serine? That would just be a ester. Why? Because the side chain of serine is a alcohol. Okay, with this one, it's the N-terminus of glycine, so that's why we have a amide. Because you're using the alpha amine on the protein to form the bond with the fatty acid. And then this one right here, we're using this sulfur right there to form a sulfur carbon bond right there. So that is just a, not a thiol, that would be a sulfide. So that's a sulfide bond. Okay, so that's the first three. That now we have this guy right here. So maybe we should clean, well, let's just use a different color pen. So look at this protein right here. The previous three, we made life easy. We just take a protein, we attach a fatty acid, then it inserts into the membrane. Here, we have the protein of interest, and now you have all these sugars right here. And now you have some fatty acids here you can see that are within the membrane. But when you look at that molecule, this whole thing right here, I want you guys to look at that very carefully. Can you find the glycerol in that molecule? So I would like you to pause the video and tell yourself, where is the glycerol in this setup right here? So the glycerol molecule is going to be right here. There's the 3 carbons with the 3 oxygen atoms attached. There's our glycerol. Okay. Now this protein right here is going to be linked via GPI anchor. And that is going to be bound here. So let's figure out what the GPI stands for. Let's write that down. Let's clean this up. There's a lot going on this slide. Okay, so GPI. Glycosyl phosphatidyl inositol. Okay, that's the linkage that we're going to anchor this protein, or that's the name of the linker to anchor this protein to the membrane. Now this linker, you can see, is drastically more complex than the previous 3. So there's a lot going on here. So let's break down its parts here. Okay. So let's talk about how this right here, that part right there, because what it's doing is that this right here is the C-terminus of our protein. So our C-terminus would look something like this. All right? And now what have we replaced that C-terminus with? That piece right there. So it would look like this. Now we would have our NH and then our CH2, CH2. And then that's obviously attached to the phosphate right there. But it's really important to understand that this linkage right here is by this amide bond right here at the C-terminus of the protein. This linkage right here should be very familiar to you based off of the head groups that you've seen in our discussion with the fatty acid, or the head groups that are attached to our lipids. So you see that's the ethanolamine head group that's forming that linkage right there. All right. So what do we have so far? Some things that you can relate to. This is the glycerol. That's our ethanolamine piece. Now what else can we say that looks familiar here? that you've seen before. Now look at this. If we take the ethanolamine and we put it to the phosphate group right there, you've seen this word before. The phosphoethanolamine, which was another head group in our lipids. Okay, so I'm hoping you're seeing all the bits and pieces of things that are familiar to you. Okay, and so all of this, okay, what we can say is, if let's use a different color here, all of this right here, all of that, even that part right there, that's the GPI linker. And I just wanted to go through that linker and show you the bits and pieces of things that you are already familiar with. Okay. So we have GPI linkers, and you can see that we have all these sugars right here. Okay. So when you come back to our slide right here, you see those sugars right there? That's kind of what we're representing now right here. Now, some other tidbits of information that you'll find useful here. Okay. And that is, where are these things found? Okay, so this guy and this guy right there are only found on the systolic side, okay, or the inside of the cell. This group right here, that can be found on both sides of the membrane. And then the GPI linkers right here are only found on the outside face of the membrane, or the extracellular space. Okay, so that's the information that I want you guys to understand about these lipid-linked proteins. Let's see, what's the next slide going to show us here? Okay. There's actually one more thing that I want to talk about in regards to the GPI linkers. And that is that this linker is a derivative of a lipid that we have seen. Phosphatidylinositol. And that is abbreviated PI. And that is just simply a lipid. So if we go back in our slides here, let's see if we can find it. Okay. Right there. So the GPI linker is just a derivative of that lipid. So if we come back here, where's that component of that lipid? You'll find that it's this piece right here. Minus that little piece right there. But it's that piece right there. That's the phosphatidylinositol. But what's but this GPI linker is a derivative of the phosphatidylinositol. And so what's different? It's the other parts. Okay. So we've attached all that sugar. Okay. We're adding that now to the PI. And then we're adding this piece right there. So I think that's really cool that this linker that's going to be very prominent in biochemistry is just a derivative of the already natural phospholipid there. So if we come back here, these linkages right here, these three, are just fatty acids stuck to a protein, and then they get inserted into the membrane. Whereas the GPI linker is a protein that's technically linked to an actual lipid. Kind of cool. Now, you know how I mentioned that these linkers are not enough to make sure that the protein doesn't just fall off and float away, and that it's because the protein itself interacts with the membrane? Well, let's delve into that a little bit deeper. Okay, let's just draw ourselves a membrane. Okay. And we know that on the outside right here is the head group with the tail like that, right? That's what's going on. So we have a polar head group on both sides of that membrane. Now, when you go back to the lipids and the fatty acids and all that, well, let's talk lipids. The head group, a lot of them have what type of charge? A lot of them are going to have a negative charge to it. Well, think about it. I mean, there's a lot of lipids that are phospholipids, so they have that phosphate group that's going to be negatively charged at physiological pH. So what's cool is that when you have these GPI linkers, let's just represent the GPI linker like that, and then you have your protein floating around, okay? That's attached to that GPI linker. What type of sequences do you think you will want in your protein in order to interact with the head groups on the membrane? And so if you have this protein keep winding around and now it gets really close and interacts with the membrane, the amino acid sequence that is designed to interact with the membrane to help anchor the protein there, the amino acids you would envision would have some positive charges to it. Right? You would have positive charged side chains on those amino acids so that they could interact with that negatively charged membrane. Now, you can't say every single lipid is going to be negatively charged. It depends on the head group. But a lot of the lipids and membranes do have negatively charged head groups. What time do we have here? So when we take a look at integral membrane proteins here, we can see this protein has what? 1, 2, 3, 4, 5, 6, 7 alpha helices right here. And it's going through the entire membrane, both leaflets right there, right? And so what type of protein could we classify that very colorful alpha helices with? That would be the polytopic. So we have the polytopic integral protein right there. And when you're looking at that protein, you see that you have some of the protein on the cytosol side, and then you have some of the protein right here on the extracellular space, and then you have a lot of the protein within the membrane. So using your intuition, what type of amino acids would you generally find that go through the membrane? You would expect to find a lot of the hydrophobic amino acids. Whereas the part of the protein that is on both sides of the membrane, where it's going to be around a lot of water, you would assume that a lot of those amino acids are hydrophobic or hydrophilic. Makes sense, right? Another example here is the maltoporin right here. And so you see a beta barrel. And so what is the purpose here of this beta barrel here? Well, it's basically it's creating a hole, or technically a channel, where things can go through. And channels often are very specific for a particular substrate. And maltoporins right here are going to select for polysaccharides to allow polysaccharides to enter the cell. So the major point that I want to make with this slide is when you look at other integral proteins right here, it's just we're representing it as a blob. And I want you guys to understand that these blobs right here have secondary, tertiary, and quaternary structure. And that's what this slide that we just came from is illustrating. It's not just a blob and just randomness. It is actual structure. And based off of the structure is going to allow for selectivity and for its function. Very, very important. Okay, so this slide right here, so you know how when we were looking at the lipids composition in a membrane, that the top leaflet and the bottom leaflet will have different composition of lipids, and we called that asymmetry. Well, the same thing with integral proteins is there's going to be typically asymmetry. And what I mean by that is that if we label this end right here as X and this end as Y, and I draw myself a membrane, we see the way I've drawn it is that we're going to basically have X right here, Y right there within our membrane. Let's make sure we understand that this is our membrane. All right, so it goes through the whole membrane, and we have X on what? The outside, and Y on the inside. Generally speaking, these integral proteins are asymmetric in the fact that X is always, for this particular protein, X will always be on the outside and the Y will always be on the inside. Always, always. You will not have the opposite. You will not have this same protein go through like this. Because these proteins are designed to have particular functions. And so this part is designed to function on the outside of the cell. And if you take that design and put it on the inside of the cell, it's going to be in a foreign environment. It'll be like, I don't know what to do. And so generally speaking, transmembrane proteins are asymmetric. They have to go into that membrane in a certain way in order for them to be functional. Now, I'm pretty sure you can always find an exception to that, but in my class, I want you guys to understand. That's what I want you to understand, that generally that is a true principle in how it's going to work. Let's see, physical properties. Okay, I think this will be a good place to stop, because as we get into physical properties, we're going to start be looking at some math here. And so we'll stop here for right now. And as always, if you have any questions or concerns, please reach out. I'll be more than happy to help.